Immune response to parasites. Parasites are organisms that live inside a host of another species and grabs its nutrient from the host organisms. There are different types of parasites. Protozoans are a type of parasite. They are unicellular eukaryotic organisms that causes many diseases like malaria, African sleeping sickness, leishmaniasis, Chaga's disease, etc. The type of immune response against protozoans depends on the location of the parasite. In their life cycles, they exist as a free form in the bloodstream for some time. At such stage, the humoral immune response plays a vital role. In some part of their life cycle, they also exist as an intracellular parasite where the cell-mediated immune response are most effective. The other group of parasites are the parasitic worms or helminths. Helminths are large multicellular organisms that reside inside a host, but they usually do not multiply and they are not intracellular pathogens. Since their numbers are very few in a host, the host immune system is not fully engaged in responding to the parasite. Parasitic worms causes varieties of diseases in both human and animals. A parasitic worm, Ascaris, infects more than 1 billion people in their small intestine. Immune response against protozoans. Immune response against protozoans and the effectiveness of the response depend on the location of the parasite within the host. When they exist as a free form in the bloodstream of the host, the humoral immune response is most effective, whereas when they exist as intracellular parasite, cell-mediated immune response works most effectively. Pathophysiology of malaria. Malaria is one of the most devastating diseases caused by protozoans of the genus Plasmodium. It affects about 10% of the world population and causes death of millions of people every year. One of the most virulent and prevalent species of Plasmodium is Plasmodium falciparum. Plasmodium species goes through a series of developmental stages in their life cycle. The female Anopheles mosquitoes, which feed on blood meals, serves as a vector for Plasmodium. A part of the life cycle of the parasite takes place in the gut of the mosquito. Human infection takes place when a mosquito feeding a blood meal deposits sporozoites, a stage in the life cycle of the parasite in the human host. Sporozoites are free-form stages of the parasite in the bloodstream, but it quickly disappears within half an hour of its introduction and migrates to the liver, infecting the hepatocytes. Within the liver, sporozoites multiply extensively and develop into merozoites within a week and later release. It is estimated that a single sporozoid infecting a hepatocyte can release about 5,000 to 10,000 merozoites. The released merozoites then infects the red blood cell or the RBC and initiate the symptoms and pathology of malaria. Merozoites further replicates and differentiate within the RBC and release more merozoites. Later, some merozoites differentiate into male and female gametocytes, which may be ingested by female Anopheles mosquitoes in a blood meal. Within the gut of the mosquitoes, the gametocytes mature into gametes and fuse to form zygotes, which multiplies and differentiate into sporozoites. Immune response to Plasmodium infection. Children below 14 years of age have low immunity to Plasmodium infection, which is demonstrated by their low serum antibody against the sporozoite stage of the pathogen. Only 22% of the children living in endemic areas have detectable antibodies to the sporozoite stage, whereas 84% of the adults have such antibodies. The pathogen has several stages of maturation where they change their surface molecules when they transform from one stage to the another. This continual change in the surface antigen slows down the immune response. Again, their intracellular life stage in the liver and erythrocytes shields them from the immune system and reduces the immune activation. Sporozoite stage is the most accessible stage in their life cycle, where they circulate in the blood as free form. However, it lasts for only about 30 minutes, which is very short to mount an immune response. Even when an antibody response does develop to sporozoites, Plasmodium has evolved a way of overcoming that response by slogging off the surface CS antigen core, thus rendering the antibodies ineffective. Therefore, once the pathogen establishes itself inside the host, it is very difficult to eliminate by the host defense mechanism. One strategy to develop vaccine for malaria consists of Plasmodium sporozoites attenuated by X irradiation. However, it is a difficult task to produce sporozoites in large scale, as sporozoites do not grow well in cultured cells, and an enormous insectary would be required to breed mosquitoes to prepare enough irradiated sporozoites to vaccinate just one small village. Immune response to parasitic worm or helminths. Helminths are large multicellular parasites that reside inside the host body. They are usually present in small number and are not present intracellularly. Since their number is few, the immune system do not respond strongly against them. Domestic animals are mainly infected by helminths, and it will be transferred to humans when they consume contaminated food or not well cooked foods. Few of the common helminths are Taenia, a tapeworm of cattle and pigs, and Trichinalia, the roundworm of pigs that causes trichinosis. Pathophysiology of Schistosomiasis. One of the common helminths is the Schistosoma species, which are responsible for a chronic debilitating and sometimes fatal disease called Schistosomiasis. There are three common species of Schistosoma: Schistosoma mansoni, Schistosoma japonicum, and Schistosoma hematobium, which are the major pathogens in humans, infecting individuals in Africa, the Middle East, South America, the Caribbean, China, Southeast Asia, and the Philippines. Recently, there has been rise in incidence of schistosomia due to increased irrigation, which expanded the habitat of freshwater snails that also serve as hosts for the pathogen. Infected snails carry and release large number of free-swimming schistosome larvae called cercariae. Infection to human occurs when they are exposed to these infectious cercaria. An infected snail can release cercaria at the rate of 300 to 3000 per day. When cercaria contact human skin, they secrete digestive enzymes that help them to bore into the skin, where they shed their tails and are transformed into schistosomules. The schistosomules enter the capillaries and migrate to the lungs, then to the liver, and finally to the primary site of infection, which varies with the species. S. mansoni and S. japonicum infect the intestinal mesenteric veins. S. hematobium infects the vein of the urinary bladder. Once established in their final tissue sites, schistosomules mature into male and female adult worms. The worms mate and the female produce at least 300 spiny eggs a day. When these eggs are released, the infected individual started showing symptoms of schistosomiasis. These eggs invade the intestinal wall, liver or bladder and causes hemorrhages. A chronic state can also develop in which the adult worm persists for a long time and the unexcreted eggs induce cell-mediated delayed type hypersensitivity reactions resulting in large granuloma formation. Formation of granulomas in the tissue causes massive tissue necrosis due to the lactic enzyme released by the macrophages. Although an immune response does not develop to the schistosomes in most individuals, it is not sufficient to eliminate the adult worms and the worms can survive up to 20 years. The schistosomes would appear to be the form most susceptible to attack, but because they are motile, they can evade the localized cellular buildup of immune and inflammatory cells. Adult schistosome worms also develop mechanisms to evade the immune response. They can decrease expression of surface antigens and can also form protective seal around them with glycolipids and glycoproteins derived from the host. The common antigens observed in the worm are mostly ABA blood group antigens and histocompatibility antigens, which are treated as self-molecule by the host. Immune response to schistosomiasis. The major immune response to schistosomiasis is a humoral immune response characterized by increase in IgE antibody against schistosomes. The other response are increase in mast cells, their degranulation and increase in eosinophils. This manifestation suggests that cytokines produced by a T helper 2-like subset are important for the immune response. Interleukin 4, which induces B cell to class switch to IgE production, interleukin 5, which induces bone marrow precursors to differentiate into eosinophils, and interleukin 3, which along with interleukin 4 stimulates growth of mast cells. Degranulation of mast cells releases mediators that increases the infiltration of such inflammatory cells as macrophages and eosinophils. The eosinophils express Fc receptors for IgE and IgG and binds to the antibody-coated parasites. Once bound to parasite, an eosinophil can participate in antibody-dependent cell-mediated cytotoxicity or ADCC, releasing mediators from its granules that damage the parasite. One eosinophil mediator called basic protein is particularly toxic to helminths. In conclusion, there are large number of parasites that causes many diseases in both human and animals. The two major groups of parasites are the protozoans and helminths. They reside within the host and use many mechanisms to survive and infect them. The type of immune response differs with the species and the type of invading pathogen. In case of malarial parasite, they have various life stages within the host, and this is a strategy to survive and establish themselves. Such infections are very difficult for the host defense mechanism to eliminate from the system. In case of infections by parasitic worm or helminths, they usually are present in very few numbers and they do not evoke a strong immune response. Therefore, they can survive for a long time within the host, causing debilitating chronic infections.